Okay, so now we start session 1 on advanced manufacturing, and our first speaker is Professor Maria Farsari, and the topic of the presentation is 3D auxetic metamaterials as scaffolds for tissue engineering. Now let us listen to the biography of the speaker from Daniel.
Absolutely. So, Professor Maria Farsari is a research director at IESL, where she joined in 2003. Her main research interests are laser-based additive manufacturing, multi-photon lithography, laser-based fabrication of 2D and 3D micro and nanostructures, and materials processing using ultrafast laser. Maria received her undergraduate degree from the physics department, University of Crete, and her PhD in 1997 from the physics department, University of Durham in the UK. After graduating, she worked as a postdoctoral research fellow at the University of Durham and Sussex, and as a senior optical scientist for the security company DeLaRue Holographics. She was a founding member of the Dublin company Xsil Ltd.
Okay, thank you very much, Daniel. So now let us welcome our speaker, Professor Maria Farsari, to give her invited talk.
So hello, everybody. I hope you can hear me. Let me start sharing my screen. Can you all see my presentation now? Yeah, so hi everybody, and it's a real pleasure to speak to you. Thank you. I would like first to thank the organizers for inviting me and accommodating me in time, since I have to leave for traveling soon after this talk. So I'm going to be talking to you about our latest work on 3D auxetic metamaterials as scaffolds for tissue engineering. And since I understand tissue engineering is not too close to a lot of these people, to a lot of the people attending this talk, I will give you a very, very long introduction. So a few things about Forth. Forth is in Crete. Crete is the most southern place in Europe. And a lot of people think it's the tropics, but it's not. We have big mountains, as you see in the photographs, and we have a lot of snow. I can see the snow outside my window now. And Forth is the biggest non-Athens-based research institute in Crete, and next year we're celebrating our 40 years of existence. Let's go now to my talk, and why do we want to work with tissue engineering? The truth is that there isn't enough there isn't enough organs to give away to the world when something goes wrong. Usually when somebody gets very ill and he needs to replace an organ, we have to wait for somebody to die to get it. Or you have to wait for a live donor, which is usually a relative. And none of these are particularly pleasant. And also there's another negative aspect to this, because it's the new organ is a foreign body inside the host body. The person who received it has always to take immunodepressant drugs so that they don't reject this organ. So there's a newish field. It's been around for about 35 years, called tissue engineering, and the idea behind this field is to try to grow organs rather than replace them with donor organs. The idea is that you take some cells from the person who is ill. So this using their own cells gets rid of the problem of rejection. Then you multiply those cells in the lab, and you differentiate them the way you want them to differentiate. Then you put them on a scaffold, which is suitable for the organ you want to make. It can be a hard scaffold or a soft scaffold. Then you grow, and once it grows, then you can transplant it to the person that needs the organ. This way, you don't need don't need a donor.
Professor Maria, sorry to interrupt. So I think we are still seeing your first slide.
The introduction of fourth. What is the slide you see?
The first slide, the opening slide with your title. So that's the slide we are still seeing. Are we in the first slide?
Yeah. That's not the one I see though. I don't know how we can.
And it's not in the, I think, presentation mode. It is still the other mode.
The way I see it in my computer, it's in the presentation mode, and I see the third slide. You still see the first slide now?
Yes, we still see the first slide.
I don't know what to do.
I think at the bottom of your page, there is a slideshow option. So I think it is already.
No, but I am on the slideshow option.
Maria, maybe you can reshare screen, stop sharing, and then start sharing again, and try to select the one you want.
Because I can see my presentation moving normally. Sorry about this. Stop sharing, and I will share again. What do you see now?
We see the fourth slide.
Now, did you change?
We still see the fourth slide.
You should be able to see number 3 now, because I see number 3.
Currently, it is, I think, still fourth slide, scaffolding.
I don't know why you cannot update it, because I can see it. Now it's not changing.
No, it is not changing. So I think we had a similar problem with yesterday. I mean, with this, and I'm not sure how it got fixed.
If I may, I believe if you stop sharing and then you reshare, but you reshare going share your screen, not sharing the presentation, I think it then should work.
Yeah, probably. Yes.
Stop share, okay, and
look for what it offers you to share.
Okay, yes. So it is share screen. I press and then it comes up to a lot of things that I have open, and I put PowerPoint presentation.
Yes. Go instead of going PowerPoint presentation, go share screen.
No, but this is part of the share screen thing, and it tells me at the top your screen sharing.
Correct, but so when you go into the presentation and you make the presentation mode, that's then a separate window from the presentation. So in order to fix that, you need to go just share screen. If it's screen 1 or if it's screen 2, it should be screen.
I'm gonna try to remove my external screen. Maybe that will help things.
It did, but now it's on. If you now reshare it to your screen 1, it should work.
Okay, so maybe that it is because I have 2 screens here. Let me, but I need use slideshow.
There we go. That's it. Perfect.
Yes.
Okay. Okay. So, okay, it's never happened before. But anyway, so I've discussed this slide. What I wanted to say at this slide at the end of the conversation was what our research is focusing is on the scaffold fabrication. Now, scaffolds, did you see my screen? Do you see my scaffold? Okay, great. So now the scaffold of the body of tissue is a very, very important structure. All cell, firstly, the body is a very complex structure. It has lots of different cells, and all these cells reside in a scaffold, which is called the extracellular matrix. So when you want to make this scaffold, you really want to be able to try to imitate this extracellular matrix and its function. Now, the research in this field started in the 90s, and it was all done flat at the beginning because 3D printing didn't exist. Then 3D printing started developing, and people started making kind of boxes where like cube structures where they with specific periodicity, which they would grow cells on, and they would see how cells grow. And this is fine because this was what the capabilities of the technology were then. But the reality is that no tissue in the body is square. We don't have this kind of periodicity that the original 3D printed structures made. And so there's a new trend now, and we are involved in this, to use metamaterials as scaffolds. So materials who have kind of designer properties. This gave, so this is what metamaterials is, mechanical metamaterials are structures whose properties are not defined by the material they are made of solely, like most materials, but they're also fundamental to the architecture. The architecture is fundamental to it. And they have, mechanical metamaterials very often have properties which we don't normally associate, like strength and low density. I mean, a class of metamaterials is the electromagnetic metamaterials that a lot of people have been working on for a long time. So there is a new field called biometamaterials, which are mechanical metamaterials with biomimetic properties. The aim of our work and what I'm presenting here is the design and engineering of scaffolds that can home cells and direct their functions and expand the forms of mechanical signal in a reproducible way so that they can be used in the fabrication of advanced scaffolds to precisely tune their biological behavior. Now, one of the major classes of materials that we've been working with is auxetic metamaterials. Auxetic metamaterials are basically materials that have negative Poisson ratio. They are used extensively in technology. A lot of helmets and a lot of knee pads are made using foams. But one of the reasons that we want to use them as scaffolds, the main reason basically, is that there is a lot of theoretical work done to say that when auxetic metamaterials facilitate mass transport better than non-auxetic materials. And this gives us the potential to have the optimal delivery of nutrients, metabolic waste, and therapeutic agents. And this is much better than any non-auxetic structure for scaffolds. Of course, there's other potential applications in biology, like stents and skin grafts and artificial blood vessels. They're very, I mean, auxetic materials have been used extensively in stent fabrication because of the way they can expand inside the veins, and also they can, when they break, they kind of break inwards. They don't injure the veins. Now, the material, how do we do that? We are using multi-photon lithography to fabricate the structures. Professor Giotkes has already talked about this, so I'm not gonna expand. So basically, it's a direct laser writing technique, which allows us to directly write 3D structures inside the material with nanoscale resolution. The materials we like to work with are organically modified ceramics. Now, the reason we like to work with them is that because they have very good mechanical and chemical properties, stability, but also because we know how to tune their properties. We can mix different inorganic materials to make to give them the properties we want. And we can also easily functionalize them in the bulk and the surface. The material we use mostly, and which I'm going to be talking to you about today, is a material we developed with Boris Cheskov's group about 20 years ago now it is. It's a zirconium silicate, which has very good material properties, and it doesn't, one of its main benefits is that it doesn't shrink during processing. So when we design a structure, we can directly translate to the design to the 3D printed structure without any compensation. So moving now to the main part of my talk. And so the idea is, the question we want to answer is, can we influence cell development and differentiation using topography and not chemistry? To do that, we chose two completely different geometries. What you see on your left is a non-oxidic geometry called Kelvin foam. Kelvin foam is very well known because it's supposed to be the ultimate strong structure. It has the best ratio of weight to volume. Basically, it's extremely strong structure and very light. Also, it is extremely stiff. So this is the one end of our studies. The other end is an oxidic environment. We also use the classic geometry here. It's called the area-entrant honeycomb structure, or also bow tie structure. It is easy to make it using 3D printing very reliably, and it also has the property of being oxidic. It has a negative Poisson's ratio. Another difference between the two geometries we chose is that the Kelvin foam structure is kind of has a kind of center of symmetry, while the Kelvin foam, the oxidic structure doesn't. So there's some mechanical modeling, and I hope you can see what happens when we put a pressure on the structures. This is what happens to the oxidic structure, and this is the top view. And you can see how the weight is distributed. While if you look at the Kelvin foam, there is really no deformation when we apply pressure to it. The kind of weight is distributed more or less evenly. Now, for the mechanical testing, we used some very specialized equipment that exists at Berkeley University. It's a kind of nano intender in an environmental SEM. And it gave us some very nice pictures and very good idea of what's happening in when we apply pressure to these structures. You see that when we have an oxidic structure and we press it, it really kind of deforms only inwards. Now, these structures, the ideal way to characterize the structures is to have them freestanding, but they're very small and they're very difficult to handle in an SEM without having an anchor point. So this kind of affects our measurements. It's like a Schrödinger's cat thing, but that was the only way we could really do the measurement. So we also tested the pore size. Pore size is very important, and there's a lot of work finding the ideal pore size for every cell type. There is no universal good number for this. What this studies give us is that by the oxidic structures, they're not just only oxidic, but also extremely deformable and adaptable. And this is very important when you're trying to grow tissue, to have on one side to have the cells contained to a specific area, and this is what this kind of scaffolds give us, but at the same time give the cells the possibility to change the topography, the interior topography. And we are doing a lot of imaging work now to see exactly how this topography is affected by the cell growth. Now, there is some interesting results when you do the cell penetration analysis. What we see is that the auxetic scaffolds allow penetration, and they are they're everywhere basically, but more or less the same thing happened with the Kelvin foam structures. There wasn't any significantly structures. There were not any statistically scientific deviations. What there was a specific deviation was the alignment of the cells, and aligning the cells is something that the biologists really want to do, because this is how they get the cells to connect and get specific properties. What you see here is how on a Kelvin foam, which has a center of symmetry, the cells went had a kind of random distribution of orientation, while in the auxetic scaffold, there is very, very specific. The cells seem to follow the axis of symmetry of the auxetic scaffold. What happens now if you put the cells in an osteogenic medium? You see here the two different geometries, and the top is the auxetic geometry. At the bottom is the non-auxetic geometry. And what you see is that the cells grow very well, and they continue to grow very well. And what you see, and it's not obvious to the non-trained eye, is that at the top of at the auxetic medium, we start to see some calcium. So what this means is that after 14 days, the cells start to produce bone. And we see this with a technique. The biologists see this with a technique called alizarin staining. And this is where the real breakthrough with the auxetic scaffolds is. What you see is, and it's obvious on the top right photograph, the auxetic scaffold and the Kelvin foam next to each other, and the control next to it. Aliresin is a calcium staining. In most symmetric or classic cultures, you can start seeing this red staining after 4 weeks. When we're using auxetic scaffolds, we have halved this time. It goes down to only 2 weeks, where we can see real calcium production and real bone regeneration. And this is also confirmed by some more tests. RANX2 protein is a mineralization protein, is a bone regeneration protein. And what you see is that when we're using auxetic scaffolds, we have several times higher RANX2 protein expressed than when we use any other type of scaffold. So the question now we need to answer, and we're still trying to debate this before we publish the paper, is, are auxetic scaffolds better scaffolds? We know that in theory they are. We know that theoretical studies tell us that we have better nutrient penetration. But what we cannot distinguish yet is if it is what makes the difference is the oxycity of the scaffolds or the elasticity of the scaffolds. One of the things that the auxetic scaffolds allows you is this adaptability of this free movement of the cells inside the scaffold. And what we are trying to do now is design some kind of elastic scaffolds using, again, SZ2080, the same material, which, however, are not auxetic. And we will try to distinguish whether it's the oxycity of the scaffolds that gives us this superiority, or is it just the fact that the cells have the space to adapt and grow. So to conclude, I didn't talk much about multi-photon lithography because Professor Zlotkas spoke before, but what I wanted to say here is that it is a technique suitable for investigating materials and architectures for applications such as scaffolds for tissue engineering. Materials engineering is crucial and for the development of tailor-made devices and scaffolds. Architected materials can provide new routes for the development of functional adaptable scaffolds. And auxetic scaffolds can induce early osteogenesis. I would like to finish by thanking my group. This is who we are. A lot of this work was done at Berkeley, but the group of Professor Kostas Grigoropoulos and his former student Zacharias Vangelaitos, who's moved to Apple now. And of course, very significant in this work was the work of George Flamourakis, my former PhD student, and Arthi Ranella, our biologist. And this is, I would also like to help our funders, which are mostly from the European Union. And thank you for listening.
Yeah, thank you very much, Professor Maria, for the fascinating presentation. So I think we learned a lot about your research. So now let us, see if there are any questions from the audience. Any questions or anything in the chat window? Okay, so there is a question from Professor Solis.
Thank you for interesting talk. So can it be that real breakthrough will be that your bones will be regenerated and they will be stronger than before in the final application?
The stronger than before it's something that we cannot determine now because how
it's
strong
obvious,
is the bone? However, what
you will not break them.
No. Firstly, what we wanted at the event when we are implanting the bone is that we wanted to have a fully grown bone. Now, what we know now is that we can grow bone much faster than traditional methods. And really, people go into a lot of effort to reduce the time of bone growth by 1 or 2 days. What we have done is we halved this time, which is really significant. And this could be partly because of the auxetic behavior. I think we also need to exclude elasticity. We do not know how much a factor it is in this. My belief is that it is, but it's not the only factor. The fact that you have, because other people have used elastic scaffolds before, and they haven't seen this kind of regeneration. I think it's a combination of the this kind of adaptability that the cells have with the fact that nutrients can travel much better in an auxetic material that gives us this result. Now, whether this bone will be stronger than before, this will depend on the cell donor. Every person has their own kind of genetic marking, and when you're using autologous grafts like what we want to use eventually, whether the bone will be good or bad will depend on what the cells that we used were. So it's not the answer is that it's not going to be any better. The person is going to be, I don't think it's going to be any healthier than it was before it had the injury, but it will not have the injury.
Good, thank you.
Yeah, thank you very much. And we have a question from Professor Mangir Das.
Thank you, Maria, for a great update. My question is related to: do you see a benefit from making combining with calcination, yes, to making harder scaffolds, which could also be auxetic? Is there any use of it, at least for some specific, like ultra stiff materials, but at the same time flexible ones?
The honest answer is that I think calcination is going to be the only way, because you really don't want to put in a real scaffold, you don't want to put organic materials that are not biodegradable inside the body. What I could see happening once we have proved this process is that to move to calcinated scaffolds and repeat the same work. However, in this case, we will not have all these acrylates, methacrylates, and all those nasty things, which eventually you would hope that you would get rid of, but it's better if you don't have them from the beginning.
Okay, thank you. I like this answer a lot.
Yeah, and I mean, most people that are doing classic work with bone scaffolds, they're working with minerals. So that's what we want to work. Eventually, we want to work with minerals.
Great.
Okay, so I have a question, Professor Maria. So is it possible to make, say, flexible, stretchable devices using your techniques?
Flexible due to geometry or flexible due to the material?
Material, yes.
There are some PDMS type materials which we can process. And in fact, PDMS can be processed with 2-photon. So the answer is yes.
Okay, great. So it can be used for, say, for example, a flexible diffraction grating where we can stretch it and change the diffraction angles.
Yes, I think this, we have done very little work with PDMS, and that was many years ago, but there is a few publications on this subject on printing, on multi-photon printing of PDMS and PDMS type materials.
Yeah, thank you very much. So let us see if there are any further questions. Okay, so there are no further questions, but would you kindly show your second slide, because you talked about your institute and then we missed that slide, I think.
Okay. Sorry, this was a first time for me to know that if you're using an external screen, it messes up your. So.
Oh, okay, nice. Yes.
So this is where we are, and the institute is in the countryside, in fields of olive groves around, but at the back we have the very high mountains. There are 2,500 meters, and we get really a lot of snow. People get very surprised when they come here. They think Crete is in the tropics. It's not.
Yeah, thank you very much, Professor Maria.
It's very beautiful. It's in the same time zone as Estonia as well.
Yes.
It looks far away, but it's on the same.
Yeah, it was very easy to plan this presentation. So thank you very much for everything.
Yeah. Thank you very much for your talk.